Hello everyone. The TI plasmid of Agrobacterium tumefaciens is a natural vector for the genetic engineering of plants. However, the wild-type TI plasmid cannot be utilized as the general gene vector. In this module, we will discuss about the methods of development of the TI plasmid into binary and cointegrate vectors. The learning objectives of this module are features of a gene cloning vector, cointegrate vectors, binary vectors, properties of binary vectors, improved binary vectors. The features required in a typical plasmid vector include an origin of replication by which multiple copies of the plasmid can exist in a bacterial cell, a multiple cloning site consisting of unique restriction sites, a selectable marker, generally coding for antibiotic resistance, and a screenable marker which can help in distinguishing transformed from the non-transformed cells. The tDNA based vectors must have tDNA borders. All other features of a plasmid vector, which are of two types, cointegrate vectors and binary vectors. The cointegrate vectors are produced by integrating a recombinant E. coli plasmid containing the gene of interest into the TI plasmid. A recombinant plasmid is generated in E. coli, which is then mobilized into Agrobacterium containing a disarmed TI plasmid carrying a part of the E. coli plasmid DNA within the tDNA borders. Generally, the tDNA oncogenes are replaced with sequences from E. coli plasmid along with the selectable marker genes for both Agrobacterium and plants. Co-integration of the two plasmids is achieved within Agrobacterium by homologous recombination. For the homologous recombination to occur, the E. coli plasmid and the disarmed TI plasmid must have some sequences common to both. For the transfer of the recombinant plasmid from E. coli into Agrobacterium, a process called triparental mating is followed. An E. coli strain having a helper plasmid carrying genes for broad host range conjugal transfer is used. The three bacterial strains, that is the E. coli strain having a helper plasmid, the E. coli strain carrying the DNA insert as recombinant intermediate vector, and Agrobacterium tumefaciens carrying the disarmed plasmid are mixed together. The conjugation between the two E. coli strain results in the transfer of the helper plasmid into the E. coli strain carrying the recombinant intermediate vector. Then the helper plasmid induces the conjugal transfer of recombinant intermediate vector into Agrobacterium. The Agrobacterium contains both the recombinant intermediate vector and the disarmed plasmid. Homologous recombination then transfers the gene of interest into the tDNA region of TI plasmid by homologous recombination as seen earlier. The intermediate vectors used for the development of cointegrate vectors has resulted in the development of a large cointegrate plasmids. But this large size is not necessary for the transformation, as this large size involves tedious and cumbersome handling procedures during cloning. In the year 1983, Hakama et al. And Deframond et al. Determined independently that the two main components for successful Agrobacterium mediated gene transfer, the Vir and the tDNA regions of TI plasmid, could be split into two separate replicons. A typical binary vector has the tDNA borders enclosing a selectable marker into which a multiple cloning site is inserted. In addition, there is a screenable marker or reporter gene. The selectable marker and screenable marker genes are driven by eukaryotic promoters and are hence expressed only when the tDNA is transferred to the plant. Outside the tDNA region are the origins of replication for maintenance of the binary vector in E. coli of an Agrobacterium. Besides, there is a region that promotes mobilization of this plasmid through conjugal transfer between bacteria. There is also a bacterial selectable marker that is driven by prokaryotic promoter and is used to select bacterial cells carrying the binary vector plasmid. tDNA is cloned on a small E. coli plasmid which is called as mini Ti or micro Ti. The DNA is integrated within the tDNA region of mini Ti to produce recombinant mini Ti. This recombinant mini Ti is introduced into an Agrobacterium strain carrying a Ti plasmid from which the tDNA has been removed. As explained in the development of cointegrate vectors, the tDNA plasmid is introduced into Agrobacterium by a three-way cross, that is triparental mating, or by a simple transformation procedure such as electroporation. The Vir gene products then bring about transfer of tDNA from binary vector to plant cell. The Agrobacterium strains having the helper Ti plasmids are selected by the presence of several antibiotic resistant marker genes on Agrobacterium chromosome as well as on the Ti plasmid. For example, LBA4404, which is an octopine producing strain, TIACH5 of Agrobacterium shows rifampicin resistance due to its chromosomal background. The Ti helper plasmid in this strain is resistant to streptomycin. Hence, this strain is selected on a medium containing rifampicin and streptomycin. The binary vectors available vary in the size, replication origins, unique restriction sites, and the selectable marker available for bacterial selection. For example, if the binary vector PBIN19 is introduced into the LBA4404 Agrobacterium strain, the recombinant LBA4404 will grow on kanamycin medium since PBIN19 harbors the kanamycin resistance gene. Further improvements in binary vectors have led to development of superbinary vectors that carry additional Vir genes on the vector backbone, which facilitates transformation in recalcitrant species like monocots. Another improvement in the binary vector is the inclusion of multiple left borders in the binary vector. This was done because incorrect cleavage of the Vir D1 endonuclease led to transfer of the vector backbone into plants along with the tDNA. Introduction of multiple LBs ensured cleavage at any one of them so that only the tDNA was transferred to the plant. In order to use tDNA to simultaneously transfer multiple genes to plants, the size of the binary vector had to be further reduced. This was done in the pgreen based vectors, which had a smaller sized origin of replication RPSA, and also several conjugal function genes were removed. This origin was recognized by a replicase Rep-A that was provided in trans by another plasmid PSOUP introduced into the same Agrobacterium cell. This enabled the binary vector to be maintained in Agrobacterium itself, and the small size of the pgreen plasmid allows insertion of multiple genes into the plant. The module can be summarized as: the Ti plasmid of Agrobacterium tumefaciens is a natural vector for genetic engineering of plants because of its ability to transfer tDNA from the bacterium to the plant genome. Any gene of interest inserted within the tDNA borders can potentially be transferred to the host plant. This has led to the development of vectors based on Agrobacterium tDNA for efficient transfer of desired genes into plants for various applications. Cointegrate vectors were developed first, which incorporated a genetically engineered E. coli plasmid carrying a gene of interest into the tDNA region of disarmed Ti plasmid. The gene of interest was transferred to plant cell by Vir genes present on the same plasmid. Binary vectors are small and cloning of the gene of interest is carried out in these vectors in E. coli, and then the vector is moved to an Agrobacterium strain which provides the Vir region in trans for transfer of the gene of interest into plant cells. To summarize, we can say that owing to its ability to transfer genes from bacterium to plant cells, TI plasmid can be utilized for transfer of multiple genes to the plant cells. For this purpose, TI plasmid can be developed into binary and cointegrate vectors. Thank you.